Yeah. So I am Rishabh and I am from IIT Kanpur and I am a PMRF scholar and I'll be helping you out with the problem solving in the course Plant Development Biology. So today we'll be starting with the week 1 discussion and let's get started. So just a second. Yeah. So this is the first question. Which of the following statements is false about the quiescent center? So, you know quiescent center that it is present in the root apical meristem and it is present in both primary and lateral root meristem and it is connected with the stem cells, but it doesn't have the actively dividing cells that are present in the root. So the correct answer would be that D, that it doesn't have the actively dividing cells that are present in the root. So this can be seen with this diagram that just a second. With this diagram, the quiescent center is shown in yellow color, that is been widened in this figure.
Sir,
And
screen is not visible.
oh, just a second. Is it visible now?
No, sir.
Oh. Then I must stop sharing, then I can start sharing again. So is it visible now?
Yes, sir.
Okay. So I am repeating again this question. So the question is, which of the following statements is false about the quiescent center? So the thing is that the quiescent center is present in the root apical meristem, that can be seen in the figure, and it is basically surrounded by the stem cells that are actively dividing cells, but it is the region that is not actively dividing, and that the cell cycle has been arrested in these region. And this region is also present in the primary as well as lateral root meristem. So this is like connected with both the stem cells that are responsible for primary growth as well as the lateral root growth. So the answer is that it does not have the actively dividing cells. Then moving on to the next question. So which of the following plant cells have haploid cell number? So haploid number of chromosomes, sorry. So first option is megaspore mother cell and the zygote. Megaspore mother cell is the one, just a second, there is someone in the call. Yeah, is my screen visible?
Yes, sir.
Hmm. So megaspore mother cell Yeah. So megaspore mother cell is the one from which the megaspore arises, and it is a 2n. It has a 2n number of chromosomes, while the zygote is formed from the fusion of male and female gametes. So it is also 2n. Then comes the option B, megaspore mother cell and the antipodal cell. So we discussed about the megaspore mother cell, but the antipodal cell is haploid in number because it is present in the embryo sac. Then comes the endosperm and egg cell. So endosperm is basically formed by the double fertilization of the sperm and the central cell of the embryo sac. And egg cell is haploid in number. And the last option is egg cell and antipodal cell. So yes, the option D is correct because both have haploid number of chromosome. This can be seen from this embryo sac image that these are the antipodal cells that are present in the embryo sac. This is the polar nuclei or the present in the central cell that after fusion with the sperm forms the endosperm. And then comes the egg that is also haploid in number, and after fusion with the sperm it forms the zygote. And here comes the synergids that are also haploid in haploid that also have the haploid number of chromosomes. So yes, the answer is D. Then moving on to the third question, the correct sequence of events during organogenesis in plants is, just a second. Yeah, is my screen visible?
Yes, sir.
So the correct sequence of events during organogenesis in plants is, so yeah, so the options are organ specification, self-fate determination, organ patterning determinacy, or the option B, self-fate determination, organ specification, organ patterning or determinacy. Third option is organ patterning, determinacy, self-fate determination or organ specification. And the fourth option is organ specification, self-fate determination or determinacy or organ patterning. So the correct option is option B. Why? Which can be explained from the given diagram. Like if we see the seed germination, first the cells that are present here are determined to a specific organ. That's how the radicle is formed, and then from radicle this the we know that whole root is originated from the radicle. So that means it has organ specification. Then what is the pattern of its growth? Like it grows below the ground, and first which part germinate, which part originates first. So that is organ patterning and determinacy. So option B is correct. Yeah. So now comes the question 4. Which of the following is not a physical mutagen? X-ray, UV light, gamma rays, or white light? So the answer is option D. White light
Sir,
is not.
question 4, screen
Yeah.
is not visible.
Oh, yeah. Now it's visible.
Yes.
Yeah. So question 4 is, which of the following is not a physical mutagen? So we have the options X-ray, UV light, gamma rays, and white light. So the option correct is white light. The physical mutagens are ionizing radiations, basically that are X-rays and gamma rays, as well as the alpha beta rays also are categorized under the ionizing radiations. And how they work by breaking the hydrogen bonds within the DNA molecule. And then comes the non-ionizing radiations, that is the UV light. So UV rays actually forms dimers between the purine and pyrimidine. So this is how the physical mutagens carry out their functions. So now comes the question 5. So the question 5 says, in an experiment to identify the nature, that is dominant or recessive of mutant, a scientist backcrossed the homozygous mutant with the wild type parent, that is also homozygous, and followed by selfing of the F1. So the phenotype of the F2 generation was observed in the ratio 3:1, that is mutant is to wild type. So we have to find what is the nature of the mutation. So in such type of questions, the question mentions that the mutation is dominant or recessive. So if we consider the mutation dominant first, so in that case, let's suppose that it's capital R, because we have said that the mutation earlier is homozygous and it has been backcrossed with the wild type, that is small r. So the F1 will result into heterozygous capital R small r, followed by, when we self it, then what will be the ratio? This will follow Mendelian pattern, and in the F2 we will get 3:1 of mutant versus the wild type. Mutant will be capital R or capital R small r, and this will be small r. So we get a ratio of 3:1, that is mutant is to wild type. But if the mutation is recessive, sorry, so in this case, this will be the wild type capital R, and this will be the mutant small r. So when we cross this in the F1, we get a heterozygous, that will be wild type, and after selfing this in the F2, we get the same ratio capital R or capital R small r, that is 3, and is to the mutant one, that is 1. But here the 3 stands for the wild type and 1 stands for the mutant, and in the dominant case 3 stands for the mutant and 1 stands for the wild type. As mentioned in the question, after F2 generation, selfing in the F2 generation, the ratio was observed to be 3:1, that is mutant is to wild type. So here the mutation is dominant. So that's the answer. Understood, everyone?
Yes, sir.
Okay, so let's move to the next question, that is question number 6. Which of the following represents a transition mutation? Just a second, someone was waiting. Yeah, so question 6, which of the following represents a transition mutation? So like a transition mutation is a one in which same type of nitrogenous base, being a purine, so purines are adenine and guanine, or pyrimidine, so that are C or T, cytosine or thymine or uracil. So in case of a transition mutation, Yeah. So in case of a transition mutation, a purine is exchanged with a purine and a pyrimidine is exchanged with a pyrimidine. So here in the options, when G changes, G is exchanged to C, that is a purine is exchanged to a pyrimidine. So it is not a transition mutation. Then C to T, so a pyrimidine is changed to pyrimidine, hence it is a transition mutation. And a G to A, so a purine is changed to a purine, hence it is also a transition mutation. So both B and C options are correct. So this can also be seen from this diagram as well. So when adenine or guanine are exchanged, that is a transition mutation, and when cytosine or thymine are exchanged, that are pyrimidines, so that is also a transition mutation. But when either adenine is exchanged with C or T, that are pyrimidines, that is a transversion mutation, uh, and similar is the case with the other three too. So now comes the question 7. Which of the following structures is not found in bryophytes? So bryophytes are the ones that
Sir,
doesn't
question
have.
7 is not visible.
Oh, the earlier question was visible.
Yes, sir. Sir, it is visible now.
Hmm. Okay. Yeah, it's visible now.
Yes, sir.
Okay. Which of the following structures is not found in bryophytes? So bryophytes are the ones that doesn't have the vascular elements. So the vascular elements involves both the xylem or the phloem. Hence, the chloroplast will be there, cell membrane will be there, cell wall will be there, but xylem will not be there. So the answer will be D. Hence, the xylem is not present in bryophytes, and this is basically the development or the evolution of bryophytes, and it says that the bryophytes belong to multicellular sporophytes, and after that the vascular tissue evolved in plants, that is present in lycophytes, and after that, hence xylem is not present in the bryophytes. Now question 8, is it visible?
No, sir.
Okay, wait for few minutes. Now it's visible.
Yes, sir.
Hmm. So, which of the following is the correct sequence for the life cycle of an angiosperm plant? So, in an angiosperm, like it is a plant that can have sexual reproduction. So, it has a sporophyte that can undergo meiosis and produce the gametophyte that produces the gametophyte, and the gametophyte produces the gametes, that are the male and female gametes, pollen grains and the embryo sac, and after these they fertilize and produce the zygote, that produce the zygote, and that develops into the sporophyte. So the correct answer is option C. So the sporophyte, yeah, The sporophyte undergoes meiosis that produces gametophyte. Gametophyte produce gametes, and that leads to that after fertilization leads to formation of the zygote. That can be better explained in the image given. So this is the sporophyte, and it has male and female reproductive organs, that is the stigma and the female reproductive organ megasporangium, and the micro, microsporangium that contains the anther. So here they produce the microspore as well as the megaspore that represent the gametophyte, and from that the pollen grains and the embryo sac develops, and after there the fertilization zygote is formed, and zygote develops into this sporophyte. So now comes the question 9. Is it visible?
No, sir. Visible now.
So question 9 says, which cell divides into two sperm cells during the process of pollen development? So the sperm, basically the microspore develops into the uh, pollen that have two nucleus, that is a vegetative nucleus, that is called the vegetative cell, and the other is the generative cell. So the pollen, the final sperm cells are generated from the generative cell. So the answer would be generative cell. And the whole microspore develops from the microspore mother cell, and tapetum cells are basically the cells of the layer tapetum that helps in the nourishment of the pollen grains. So the answer would be generative cell, and that would be better explained with this diagram, in which the uh, microsporophyte first undergoes meiosis and produces microspore tetrad. Then this one of the microspore produces this pollen that have a vacuole and a nucleus, and after mitosis, that is unequal mitosis, this produces a vegetative nucleus as well as the generative cell. So
Sir,
this,
I have a question.
yeah.
सर इफ क्वेश्चन में वेजिटेटिव और जनरेटिव दोनों दिया होगा तो ए एंड बी बोथ, तो उसमें कौन सा क्लिक करेंगे?
Generative would be the better one, because vegetative cells do not produce the sperm cells.
ओके सर.
So here the generative cell undergo mitosis and produce two sperm cells that basically fuses with the egg, and the other fuses with the polar nuclei, forming the endosperm. So hence the answer is generative cell. Now moving on to the next question. Is it visible?
नो सर. Visible now, sir.
Yeah. So identify the pair in which the type of growth is not matched with the type of axis it takes place. So just to refresh, if there is a plant, so if it increases in height, that is called the apical basal axis. Yeah, sorry for the interruption. So if it is growing towards the growth or the lateral growth, that is the proximal distal axis, sorry, that is the radial axis. And if there are branching in the plant and it is going from here to the end of the branch, so this point is called the proximal end and this point is called the distal end. So this is called the proximal distal axis. And in case of a leaf, so there are two axes, that is the top one and the bottom one. So that these are called as the adaxial abaxial axis. So in this case, the primary growth is associated with the apical basal axis, that is increase in length of the plant. The lateral growth is associated with the proximal distal axis. So the branching and the growth of the leaves are associated with the proximal distal axis. The secondary growth, that is this one, the radial axis, is associated with the radial axis, not the adaxial abaxial axis. This is also included in the lateral growth. So the option is C. This can be well understood with this diagram. Yeah. So this is the apical basal axis that increases the length of the plant. This is the proximal distal axis, the branching or the leaf production, and this is the adaxial abaxial axis, that is the upper part of the leaf or lower part of the leaf. And one thing is missing here, that is the radial axis that increases the diameter of the plant. So now comes question 11. Yeah, is it visible?
Yes, sir.
So question 11 says, identify the statements that are true for plasticity. So it allows the plants to adjust their form and function to small scale variations in the habitat quality. It leads to physiological as well as anatomical changes in plants. It changes in the resource uptake of the plant, or all of the above. So the answer would be D, all of the above. So what happens in plasticity basically is just a second. Yeah. So as the plants are sessile, so they are very much prone to habitat change, and due to this habitat change they have to maintain their internal physiological activities. That is why if there is any change in the surrounding environment, being it the sunlight or the drought conditions, or they are not getting proper nutrients, the plant has to change or its metabolism or the resource uptake to cope up with the surrounding environment. So that is why plasticity is important. And this can be seen with the figure that in controlled climatic conditions all the three plants are growing very well, and the plant which has very low water absorption or water uptake capacity, in drier climate conditions it generally wilt. And the other two plants that are maybe slow growth, or they have more light utilization or light adaptability, that can survive well in those conditions that have more sunlight and less water. So here comes the concept of plasticity. Now we go on to question number 12.
external.
Yeah.
But here in the options we have some, for example, first option is that it allows the plant to adjust to their form and function to small scale variations in
Hmm.
habitat quality.
Hmm.
I think this should be the most appropriate because in the rest three options it involves physiological changes, anatomical changes,
Hmm.
and in the third option as well,
Hmm.
where the changes in the resource uptake of the plant. That is,
Hmm.
it again will help the plant to adjust anatomically. But for plasticity, it is usually taken as the external phenotypic change
Hmm.
that we take it. Now, how come it is possible that all of the above will refer to plasticity if I am not
Yeah,
incorrect?
you are correct. But if we see the physiological as well as the anatomical changes in the plant, they are also the phenotypic characteristics, right? As well as the resource uptake of the plant. So, if we talk that how will the plant adjust itself onto small scale variations, so it will do by having metabolical changes, physiological changes, or changing in the resource uptake from the environment, right? So basically here all the three options like B and C are just in a different way of writing of A only. So have you understood?
Yes, sir. Yes, sir. Move on.
Okay. So now moving on to question 12, it is visible.
No, sir.
No, just wait for a few minutes.
Visible now, sir.
Yes. So question 12 says a scientist designed an experiment to demonstrate that concentration of auxin. So auxin is basically a plant hormone that helps in various physiological functions inside the plant. So auxin can act as a positional signal for development of lateral roots. So he used GFP gene, that is a reporter gene, under the control of auxin responsive promoter. So auxin responsive promoter is the one that functions in the presence of auxin. So if auxin is present there, then only the promoter will function, and if auxin is not present there, the promoter will not function, and the gene under control of this promoter will not be expressed. So after analysis, it was found that the lateral root primordial cells, that are the cells that the lateral root originate from these cells. So these cells show very high level of GFP expression as compared to the other cells. So what do you infer from this study? So the first option says auxin helps in the development of lateral roots. Just a second. Yeah. So going back to the question, so if we have a cell in which we have inserted a promoter, auxin responsive promoter, and it controls the expression of a GFP gene. So if this cell has auxin, then only GFP will be produced and it will show green light in this cell. So it says that in the lateral root primordial cells, it is showing high level of GFP expression, and lateral root primordial cells helps in the development of lateral roots. So this says, the option says auxin helps in the development of lateral roots, auxin helps in the development of primary roots, Auxin inhibits the. So, as lateral roots are originated from the lateral primordial cells, the auxin high level of auxin concentration will help in the formation of lateral roots. Hence, A option is the most appropriate here. Okay. So, any doubts in this question?
No, sir.
Okay. So, moving on to next question, that is question number 13. Is it visible?
Yes, sir.
So, what is an explant? So, explant, as we know, in a plant, if we take a plant, so it has a characteristics of totipotency, and totipotency says that each and every cell of the plant can give rise to a new organism. Okay, so every tissue or every cell of the plant can be used as a stem cell to generate a whole plant. That is, we use in plant tissue culture, and in plant tissue culture, any part of the plant that we use to grow another plant is known as an explant. So, an explant is used in plant tissue culture to develop the whole plant. This is the correct answer. And all the other three are not correct. So, this can be explained with this figure, that if we take a meristem and yeah, so if we see that if we cut a meristem, then it will be regenerated, and then same is the case with any other wound in the tissue, that will also repair itself. Then if we cut a leaf and place it, then the adventitious roots will grow, and auxiliary shoot will grow, and here we can, as in seen in this figure, the organogenesis can also happen from this cut leaves if they are placed in appropriate environment. And if we see from the root, so if we cut this section out here, so lateral roots can form from these, and at the tip, that is the meristemic part of the roots, can also help in meristem reconstruction. So every part of the plant has the ability to function as an explant, and this is due to the a feature of a plant that is called as totipotency. So, moving on to the next question. Yeah, is it visible?
Yes, sir.
So, in forward genetics, so there are two terms in when we study the genetics. So, one is forward genetics and the other is the reverse genetics. So, in forward genetics, we generally go from phenotype to the gene of the organism. So, what the phenotype suggests and what is codes this phenotype, we study in forward genetics, while it's vice versa in case of the reverse genetics. So, we first identify the gene, then we study its functions. Okay, so the option says the gene is deduced from the RNA it transcribes. This can be a correct option, but not to a full sense. So now, just a second. Yeah, someone has raised the hand. Yeah, you can ask the question. Hello, someone has raised the hand. Thank you. So, so the first option says that the gene is deduced from the RNA it transcribes. Second, the mutants are used to check the function of a protein. And the third, the gene is identified for the phenotype it encodes. And fourth option, that deduce the function of the gene. So, option C is the most appropriate option here because, yeah. So, as we can see here also in the figure, in case of this forward genetic screens, the phenotype that is uh, observed in case of mutants are used to study the genetic makeup of that gene. And in case of reverse genetic screens, first we study about the gene and what the function it encodes and what phenotype it produces. So this is basically about the forward as well as the reverse genetics. So any doubts here?
No, sir.
Okay. So moving on to the next question. Yeah, is the question visible?
Yes, sir.
So the question says the region in the TI plasmid of a recombinant agrobacterium that can be used to transfer foreign gene to the host plant is, so it is not the chromosome. Chromosome is present in every bacterium, but it is not transferred to the host plant. And miRNA. So miRNA generally are not transferred while infection of a bacteria to the plant. Similar is the case with rRNA, but tDNA is transferred. So the answer is tDNA. And how it happens? So Agrobacterium tumefaciens is the bacteria that infects plants and it causes a crown gall disease. So it has basically a plasmid that is called the TI plasmid or the tumor inducing plasmid. So this plasmid is the major cause of that crown gall disease. And TI plasmid is very large plasmid and it has a region called as the tDNA. Okay, so tDNA has the genes that are responsible for the generation of that crown gall, and like basically it produces the morphogens or the hormones like auxin or cytokinin in the plant. So it inserts the gene of auxin and cytokinin in the plant that leads to generation of a huge mass of cell, and that is called as the crown gall. But and it has been exploited in the recombinant DNA technology for generation of the uh, transgenic plants. So if we consider this, so DNA containing the gene for the desired trait is inserted into the tDNA. the gene is inserted into the tDNA, and using the normal recombinant DNA technology, that is the restriction enzyme and the DNA ligase, and after that the recombinant TI plasmid is is transformed into the bacteria, and after that the bacteria is allowed to infect the plant cell in vitro, and following this the tDNA is inserted into the genome of the plant cell. That leads to the insertion of the foreign gene into the plant, and after that the plant is regenerated from this plant cell. So this tDNA is used as a vector to transfer the foreign gene, that is the gene of interest, into the plant, and from that we can generate various transgenic plants. So this is the basic mechanism, and the answer is tDNA. So any doubt here?
No, sir.
Okay, so let's move on to the next question. Is it visible?
No, sir.
Yeah. So question 16 says a gene X was active in an adult plant, but when callus was generated, the gene or the plant part was grown in tissue culture, and the callus was generated from the same plant, but the same gene X was found to be non-functional. So what can be the reason of the finding? So the option says that UV radiation might have stopped the expression of the gene X, or the disruption of gene X may be due to TOS17 transposon, or gene X might be inhibited from the water in the media, or none of the above options. So can anyone suggest what can be the correct option for this? So yeah, so the correct option is option B. That is, the gene might have been inactivated by TOS17 transposon. Why this? Because TOS17 is a retrotransposon, and it is generally activated in tissue culture. So yeah, so if this is an adult plant, there is no expression of TOS7. There is expression, but it is not active. TOS17 transposon is not active in an adult plant. But when we grow this in a tissue culture, so due to the conditions in the culture as well as the media, TOS17 transposon becomes active. So this is the answer, and this has been explained in this figure also. So this is TOS17 and this is the gene, and if we grow this plant in plant tissue culture, and so this transposon becomes active, Yeah. So, and when we grow up this any plant from this developed culture, so in this, if we see that the gene is not being expressed because it has been disrupted from TOS17 transposon. Okay, so this is a very important concept, and you might also get the questions in the assignments as well. So, is there any doubt in this concept, or shall I explain it again?
No, sir.
Okay, moving on to the next question. Is it visible?
Sir.
No, sir.
Sir, it is visible.
So, moving forward, the technique by which the expression of a particular gene is enhanced by insertion of enhancers in the regulatory region of that gene is known as. So, there is the question says that we have enhanced the expression of a particular gene by the insertion of a regulatory gene in that region. So, this is done in activation tagging. So mutagenesis cannot be the option. Why? Because the if we mutate any region of the gene, for example, this is the gene and this is the regulatory region followed by the main structural gene. So if you mutate here, so we cannot the expression of the gene will be hampered. So mutagenesis cannot be the option. Then comes insertional inactivation. So in case of insertional inactivation, we generally inhibit or inactivate the expression of a gene. So for example, like you have seen in the TOS17 transposon also. So this is the gene, and if the transposon is inserted into this, so the gene will not be expressed, or if it is expressed, then it will not be functional. But we have to enhance the expression of the genes, that is done in activation tagging. So this can also be explained from this figure that this is an enhancer that needs to be added in a gene, and using agrobacterium transformation we use this enhancer along with the gene B that we need to increase the expression of. Okay, so we enhance the expression of this gene using the enhancers. So this is called as activation tagging. Okay, so Amardeep has asked a question. What is the LB and RB in this read in this? So yeah, in this case, this we have mentioned in the agrobacterium, right? So in that question, if we go back, right, is the screen visible to question number 15?
Yes, sir.
Hmm.
Yes, sir.
So in this question, we described about the tDNA, right? So there is a mechanism that is called as the, just a second. Yeah. So what happens is when this the Yeah. So the whole agrobacterium does not go inside the plant. It sends it sends the part that is the tDNA region only, and the tDNA is surrounded by two borders, that is the left border and the right border sequence. So how did the plant recognizes that only tDNA has to go inside the plant? So the left border and the right border are the sequences that are recognized by the bacteria, that the region, this region, only this tDNA region has to be inserted and like transfer into the plant. So this LB and RB are the left border and right border sequences that are present in the TI plasmid surrounding the tDNA region. Okay, so now moving to this. So this activation tagging is everyone understood this activation tagging?
Yes, sir.
Okay, now moving on to question number 18. So yeah, is it visible now?
Yes, sir.
So what is tail PCR is used to? So amplify random DNA fragments, sequence the region of DNA to find the insertion region of tDNA, and none of the above. So tail PCR is basically used to find the insertion in the tDNA, insertion region of the tDNA in the plant. So it is used as a marker technique to identify this region. So in that case, option C is correct. And how it's done now? So we used a set of primers that are randomly placed in the tDNA region. Okay, so these primers are not too specific, these are random primers, and one primer will be there that is specific to a region that is outside the tDNA. Okay, so we place all the primers and we set up a PCR using these. For example, this is the SP1 primer and the AD. So we set up a primary PCR using these two. Followed by the PCR product is then set up for another PCR using the primers SP2 and AD. Then followed by the tertiary PCR, the third PCR that is set up with the SP3 and AD. Okay, so here three PCR steps are done, followed by the amplified products are cloned and then sequenced. So if we have a region that is inserted into the tDNA, so this will show the specific band size as compared to if a region, if tDNA is not inserted into the plant, then no band will be observed. Okay, so this is how tail PCR is done, and this is used commonly to find the insertion region of the tDNA. Okay, so any doubts still here? Sorry. Okay, clear everyone? Shall we move forward?
Yes.
Okay, so let's move to question number 19. Is it visible?
No, sir. Sir, visible now.
Yeah. So it's an analytical question. So three plants were genotyped and it showed the following results, and the results are showed here. This is the gel image showing three bands. The position of primers are shown below. So this is the primer position that has been shown. Find out which plant is homozygous for the tDNA. So as we So as we can see in the diagram, one primer is there that is situated in the tDNA region, and the other primer is situated in the outside the tDNA region. Okay, so if the plant has the tDNA, will give a band that is equal to the band size of these two primers. And if the wild type allele doesn't have the tDNA, so it will not give any band in this region. So that is only explained in this figure. So can anyone guess which plant will be homozygous for tDNA? Yeah, anyone? Okay, plant 3. Then others? So yes, the correct answer will be plant 3. And how this came? So as we can see in plant 1, there is a band that is observed on the plant 1, the DNA region that is above this small band, this red band. So if we can see in this primer figure also, the primers that are for these genes, the for the wild type gene, so this gives us the blue band. So plant 1 doesn't have the tDNA allele. In case of plant 2, if we talk about, there are two bands observed. One is the larger band and the other one is the small one. So it says that the larger band is due to the wild type allele. But if we see the second band, so it is due to the tDNA and the reverse primer of the present in the gene of interest. So this gives us a smaller band. So hence plant 2 is also not homozygous, but it is heterozygous for the tDNA allele. Okay, and in case of third plant, if we see, if in both the chromosomes the tDNA is inserted and we place both the primers that is specific for the tDNA as well as outside that, so it will give only one single band, not the wild type one. Hence the plant 3 is homozygous for the tDNA. Okay, the question can also be otherwise, that if you observe these two bands, then what can be the phenotype or the conclusion from that? So in that case, it's heterozygous for the tDNA. Okay, so any doubts in this question? Shall I repeat this? If anyone of you have not understood, kindly let me know. Okay. So someone has messaged in the chat. So shall I repeat this problem? Okay. So what happened? So we did an essay, and in that tDNA was used to transform that plant. And now we have to check whether the tDNA has inserted into the plant, or it has been inserted in a heterozygous or a homozygous way. Okay, so uh, if we consider that we are given that one of the primers bind to the tDNA that is specific in the tDNA region, and the other primer is present outside that tDNA. Okay, so and in case of the wild type allele, there is a primer that binds to the gene that is the forward primer, and this is the reverse primer that will give us the PCR product. Okay, so if this tDNA will be inserted into this gene, so considering this is the gene of interest, and this is the forward primer, and this will be the reverse primer. Okay, and here the tDNA has been inserted, so here the tDNA specific primer will bind, and this will give a product. Okay, so this will give a PCR product, and that is shown in the red band here. And if we consider this, so here no tDNA is present, so it will give a longer PCR product as compared to this one. That is why there is a band that is above that. Okay, so if we consider plant 1 only, so it has a larger PCR product, that is it has a wild type allele. In case of plant 2, if we consider this, one of the chromosome has tDNA and the other doesn't have. So that is why two bands are observed, that is one from this, that is the wild type one, and the other from this that has the tDNA region. Okay, and in case of the third plant, if we see, only one small band is observed, that is the red one. That is because in both the chromosomes. So if we consider this is the gene, and here the tDNA is inserted, and this is the other component of that gene, and in this also the tDNA is inserted. Okay, just a second. Yeah, so the screen is visible to everyone.
Yes, sir.
Okay, so I am explaining it again because I think that some of you were not able to see my writing part. Okay, so in this case, the tDNA has a primer that is specific to this region only, and there are primers that are specific to the gene of interest. Okay, so if we consider a wild type allele, so there is no such interruption in between, so they will give a PCR product of this much length, that is shown in the blue bar. Okay, so this is a larger PCR product. No, still the handwriting is not visible. Mhm. Is it visible now? Just someone write in the text. I am writing something. Is it visible now? Is it visible now? What I am writing?
No, sir.
Is it visible now?
No, sir.
No, sir.
Not yet. My screen is visible.
Yes, sir.
Okay, it's in question 90, right?
Yes, sir. Yes, is my slide visible now? Yes, is my slide visible? Sir.
Hmm?
Yes, sir.
Okay, so now I am writing something. Tell me whether it's visible or not.
No, sir. This is simulation.
Is it visible?
No.
Yes, is it visible now?
Yes, sir.
The writing as well?
No, sir.
Is it visible or not? Okay. Just a second. Is my cursor visible to everyone?
Yes, sir.
Okay. Learning at the time. Okay. So this is the gene that have a tDNA inserted into it. Okay. So now we have to confirm whether the tDNA insertion is a heterozygous or a homozygous condition. Okay, so in that case, this primer is specific to the tDNA region only, and the other primers are specific to the gene of interest in which we have to check the tDNA insertion is there. Okay, so now if we consider the wild type allele first. So in that case, the primers that are here will give a larger PCR product. Okay, so first we are using the primers and then we will set up this condition into the PCR. Okay, so then we get a PCR product and then we will run that in a agarose gel and we'll get bands for that. So in this wild type allele, we'll get a larger band. Okay, that is represented in a blue shade in this gel. Okay, now we consider the tDNA allele, and in that case, this primer and this primer will form a product that will be somewhat smaller in size as compared to the wild type one. Okay, so now if this tDNA is inserted into the gene of interest, then this small band size will be produced in the PCR as compared to the wild type one. So now coming back to this gel image, and in this case if we consider plant 1, so plant 1 has only the larger PCR product. That means it has only the wild type allele. Okay, and if we consider plant 2, it has both the band size. So one chromosome has this wild type allele and the other one has tDNA inserted into it. That's why it's showing a smaller band size. And if we talk about the third plant, so third plant have both the has inserted tDNA into both of its homologous chromosomes. Hence, it's showing a smaller PCR product or a smaller band on the agarose gel. Okay, that is why we have this homozygous tDNA region inserted into the plant 3. That's why the answer is plant 3. Okay, understood everyone?
Yes, sir.
Yes.
Okay, so sorry for the interruption, guys, for that. And now we'll move on to the last question of today. So the complementation analysis is used for to assign mutant alleles to specific genetic loci, to find out the mutation, to carry out recombination, and none of the above. Okay, so this complementation analysis is used just to assign mutant alleles to specific genetic loci.
So
Okay,
screen is not visible.
sorry. Oh, now it's visible.
Yes,
Yeah.
Okay, so so complementation analysis is used too. So the question is why we are using complementation analysis in genetics of the plant. So to assign mutant alleles to specific genetic loci, to find out the mutation, to carry out recombination, and none of the above. So complementation analysis is basically used to assign different mutant alleles that are present in a specific genetic loci. So the answer is option A. And how it's done? So this is basically a diagrammatic representation for complementation. So complementation is basically a thing that if we are able to like if we have two genes that are coding for a single product. Okay, so if we have mutation in gene 1, then the final product will not be formed. Okay, so and if we have mutation in gene 2, then also the product will not be formed. So if we have to check whether the product we are producing, or the product plant is producing, is because of mutation in either gene 1 or gene 2. Okay, so now we use this one first. So we get a protein 1, and in this we have so in this case, if either of them is mutated and then after recombination the genes recombine and that forms a protein wild type, so this basically proves that there are different mutations in different genes and that can be complemented after their crossing. So and if there is a mutation in the single gene, like in this case, so the protein will not be produced. Okay, so then that means that it doesn't complement the wild type. Okay, so this is basically the complementation analysis. So this comes to the end of our question answer session, and if anyone has any doubt in anything that we have discussed, can ask. So you have any doubt in any of the lectures that for from week 1, can also you can also ask that also. And any of the things that we have discussed you did not understand, you can ask that also. Yeah, I'll be the team will share this material with you guys, so don't worry for that. This PPT will be available with you. Okay, Shubzar has raised his hand, so any doubt?
No, sir. No, sir. Thank you, sir.
Okay.
So good evening, sir. Good evening, sir. Sir, I have a doubt in self-fed determination, sir.
Okay.
So in that there are two types of self-fed determination, that is cell lineage based and position based, sir. Sir, how can we differentiate that lineage and position based mechanism, sir? I have a little bit doubt on that.
Okay, so uh, just a second. Yeah, are you able to see this whiteboard?
No, sir.
Then I'll explain. Yeah, sorry for the glitch, guys. Just will take us just a second.
Sure.
So my screen is visible to you now.
Yes, sir, it's visible since the question 20, sir.
Sorry.
Actually, it's visible. The question 20 only visible, sir, not the whiteboard.
Okay. Yeah. Now it's visible. There will be a
Yes, sir.
blank screen.
Yes, sir.
Yes, sir. Okay. So now I am writing something. Is it visible to you?
No, sir. So, or can you use some of the Google tools in Google Meet, sir, for writing?
Yeah. Just a second.
Yes, thank you.
Hmm. So are you able to see this whiteboard? Hello.
No, sir.
Sorry, guys. Just wait for a one minute. I'm opening that.
Sure.
As a tDNA inserted into it. Okay, so now if we use primers, we will use primers for the wild type allele, that is the original DNA gene, and the one that and a specific primer that can be a forward or a reverse primer for the tDNA that is inserted into the gene. So if we have to recognize that anything has inserted into the gene, so we have to have a primer specific for that also. So that we have here for tDNA. Okay, so now if we check into it, and let's consider the wild type first. So in that case, the prime, if we uh, PCR this, and if there is no tDNA insertion, then this whole band will be observed on the gel. Okay, so that will give us the wild type conformation. And if like in this tDNA allele, a tDNA is inserted into the gene, then this primer will go and bind to this region, and a shorter DNA segment will a shorter band will be observed on a gel. So this is basically the whole scenario that we are talking about in this problem. So that is why when there is a heterozygous, that is in one of the chromosomes, it is the in one of the chromosomes there is DNA insertion, but in the other it is not. So in that case, yeah, in that case there will be two bands observed. One will be for the wild type one, and one with which has tDNA inserted into it. Okay, and if both the chromosomes we have tDNA inserted into it, will show a single band that is shorter in size as compared to the wild type one. Okay, so we have a single band on the gel. Okay, so this was the question. Have you understood now?
Sir, if we get only a blue band, then it is of the wild type allele.
Yeah,
Means
it's
only a
only the
larger.
wild type. There is no tDNA insertion in that.
Sir, so how you are determining that means which one will be for the homozygous condition, sir? So that I want to know.
Okay, so in case of the so uh, you can just think about that there are two homologous chromosomes, right? So actually my pen is not working, that's why I cannot draw, otherwise I would have drawn. So if tDNA is inserted in both the chromosomes, okay, so we will not get the wild type band as such. It will be observed, but it will be of a very less concentration, so it will not be observed here. So only the tDNA region, that is the small band, will be observed out of it. Okay, and the larger band will not be observed. So that's how we are counting that it is homozygous for the tDNA. Okay.
Means, sir, if only the smaller band is visible, then we are saying that it is a homozygous. But if the larger is visible, then we are saying it is a heterozygous.
If larger and as well as a smaller band is visible, then we are calling it as a heterozygous. So plant 2 will be heterozygous for the tDNA because tDNA is inserted only in one of the homolog. Yeah, okay.
Sir, but in the first one, in the tDNA allele, the tDNA is inserted, but not in the wild type. So that can also be the, sir, homozygous condition for the tDNA.
Sorry, can you just repeat?
Sir, I am saying that for the tDNA allele, the tDNA is inserted there, but for the sorry.
Right, and the third plant is homozygous for the tDNA, not the wild type allele.
Yes, sir. Yes, sir. I got.
Okay.
Yes, sir. Yes.
Hmm. Sure. Anything? Any more questions?
So one question, sir.
Yeah.
So in last video of week 1 session, there is a gene tagging in which we can map the tDNA insertion site, sir. In that, there are many types like tail PCR, plasmid rescue, inverse PCR. In
Hmm.
that, every method there is one step, final step, that is cloning and sequencing, sir.
Hmm.
So in that step, how we are finding that perfect sequence of tDNA insertion site, sir?
Okay, so you might have gone through the videos, and it has been mentioned that we are sequencing, like we are doing PCR using two primers. So one will be involved in the region that is present in the tDNA, and the other primer will be in outside that tDNA. Okay,
Yes, sir.
so that's how, like, there will be some region that is present in tDNA. So we'll be determining the sequence that yes, this is present in tDNA. That is how we get the sequence of that region only, and then we recognize that yeah, this is like this confirms that the tDNA is present.
So why we are cloning here, sir? Just sequencing is here enough only, no, sir.
Yeah, actually, when we sequence the things, okay, so in like this is a general case scenario. Like in some of the vectors we have the reverse primer, like general for everything. So we just clone in those vectors only, and we have the primer specific for the region that we require, okay, and a specific reverse primer that is specific for the vector. Okay, then also we clone and then sequence. So this makes it easier for us to get that small fragment sequence only.
So we are cloning that PCR amplified product into a vector, okay?
Hmm.
So that we can sequence that vector and find out that's perfect sequence.
Yeah, actually we are not sequencing the whole vector. We are sequencing just the part that is being amplified using that, the primers of that specific primers.
Okay, sir. Thank you.
Hmm. Okay. Yeah. Any more doubts? If not, then we are closing the session for today. Okay. So, thank you everyone for being patient and listening to me. And
Thank you, sir.
Thank you very much.
thanks a lot.
Yeah, thank you so much, sir. Yeah.
Thank you, sir.
Yeah, welcome.